A nice and a favorable study time to everybody. I am Royet Puyo, and I will be delivering this week's topic discussion on the overview of your immunohematology subjects. And well, of course, in by using correlative concepts like your history, genetics, immunology, and hematology, and as well as your biochemistry for the very important RBC metabolic pathway. Beginning with the historical background and developments in immunohematology. So actually, it is for the succeeding slides, it is actually more of a presentation than a discussion since I will only be introducing you, particularly with the several milestones in immunohematology or transfusion practices. Okay, class. So in the earliest history, in year 1492, Pope Innocent VII was transfused with the blood with three young men. They eventually unfortunately, they eventually all died after the transfusion procedure. Why exactly, or what is actually the main reason why? So for Innocent the Seventh, he might have possibly died due to a rapid intravascular hemolysis, since he was probably transfused with incompatible blood types. And since class, this is the early history of blood banking and transfusion practice, the compatibility testing and the blood typing has not been discovered yet. And the three young men may have died due to blood loss. Also, since vein-to-vein transfusion hasn't been discovered, and that we can deduce that these three were caused to bled profusely to obtain the blood. Okay, imagine what I am referring to. And this was the first ever recorded blood transfusion in the history of transfusion practice, having Pope Innocent VII, the first recipient, and the three young men as the donors. So in 1628, William Harvey first described the mode of blood circulation and published this discovery in his book De Motu Cordis. So in the further development in the transfusion practice, we have first Christopher Wren in 1656 demonstrated the intravenous injection of substance into animals and its systemic effects. So for Richard Lower in 1666, he successfully transfused blood from one dog to another. It is the first animal to animal blood transfusion. And did you know, class, that our beloved dogs, regardless of the breed they have or they possess, they have only one blood type. And I don't know what that exactly means. Okay, if your curiosity arises about that certain trivia, uh, we can refer to Google, since it is not included in our textbook in Denise Harmony. So moving on with Samuel Pepys, he stated that bad blood might be mended by borrowing blood from a better body. So this postulate was actually regarded as one of the blood mythologies in the transfusion practice. So it means to say it lacks a scientific basis. So, on your first animal to human transfusion, June 15, 1667, Jean Baptiste Denis is a French physician. He treated a 6-year-old boy who had been tormented with contagious and violent fever or a disease state. He actually exchanged 3 ounces of the boy's blood of 9 ounces of the sheep's blood or lamb's blood, and he actually this is a successful transfusion. And from these concepts and perspectives and procedures, he deduced or rationalized that animal blood is purer than that blood of the humans. So next is the first malpractice suit for blood transfusion. Antoine Mauroy is a 34-year-old madman. Blood was believed to affect one's temperament and character. where blood transfusion could be used to treat mental ailments. So this is one more example of those blood group mythologies that we have. For example, taking for example your blood group B, it was first characterized that all blood group B individuals are associated with criminality, which is, of course, a blood group mythology since it does not have any scientific basis. So, as well as this one, the one postulated by Anton Mulroy, that blood was believed to affect one's temperament and character, and that blood transfusion could be used to treat mental illness, is not a science-driven postulate, which is therefore it is recorded as one of the blood group mythologies. Continuing with our first malpractice suit for blood transfusion. So Denise transfused Moroy with calf's blood. However, he complained great pains in his kidneys, stomach ache, and nausea. So later on, the transfusion was discontinued after the patient vomited and passed black colored urine, which is already a form of or a symptom or a clinical correlation to kidney damage due to the transfusion. Moving on with the first malpractice suit for blood transfusion. Uh, Moroy suffered from a relapse, and before another attempt for blood transfusion, he eventually died. The widow had been offered money from his rivals to actually charge him with murder. However, she offered to drop the matter if Denise would agree to support her financially. However, Denise refused, and this is the precise reason why the court and much of the rest of the Europe or European countries decided to ban all kinds of human blood transfusions. In 1795, Philip Singe-Physique, which is a father of American surgery, performed the first human-to-human transfusion based on a footnote in an American journal. So there is not much about this milestone in the transfusion practice. This is just an additional information. In 1816, John Henry Leacock subsequently performed and published a set of animal experiments that proved that the donor and recipient must be of the same species. So, this time or this year has actually been the year of inquisitiveness for compatibility via having a donor and recipient of the same species as a postulated theory. In 1825, James Blundell confirmed Leucas' findings that blood could be used as a treatment, but only from the same species. So, as I was referring a while ago, that the later year has been the a year of inquisitiveness for compatibility. So successfully transfused. Okay, James Blundell successfully transfused a woman dying from postpartum hemorrhage with blood from her husband. So class, take note that year 1825, by the advent of James Blundell, performed the first successful human-to-human blood transfusion. In 1840, Samuel Armstrong Lane, aided by Dr. Blundell, which is actually a colleague of Dr. Blundell, performed the first successful whole blood transfusion to treat a condition called as your hemophilia. So, everything about hemophilia will be discussed in your hematology class. Okay, understanding the basis for incompatibility. Emil Ponfic, he observed red cell lysis or hemolysis in the blood of a woman who died after receiving a transfusion of sheep blood. So, from this concept, he postulated that the blood from the sheep may not have been compatible with the blood of the woman who died after receiving the transfusion. So, therefore, incompatible transfusions were actually associated with hemorrhage and congestion of the kidneys, lungs, and liver, which is further explained nowadays, like the collapse of your kidneys from incompatible blood transfusions, and your trali, which is the main cause of fatality of incompatible blood transfusion. Trali meaning transfusion acute lung injury. So moving on with Leonard Landos, human red cells would lyse when mixed in vitro with the sera of other animals. This observation set the stage for the study of the immunologic or serologic basis of a blood incompatibility. So in 1869, Braxton Hicks recommended sodium phosphate as blood preservatives in order to prolong the viability of red blood cells while in storage um, durations. In 1900s, Carl and Stinner discovered the ABO blood group system or the ABO blood groups and explained the serious reactions that occurs as a result of incompatible transfusions. And in later year, 1901, the ABO blood group is officially or was officially reported. Moving on with Edward Lineman, he carried out vein-to-vein transfusion of blood by using multiple syringes and a special cannula for puncturing the vein through the skin. So Edward Lineman performed the first vein-to-vein blood transfusion. So moving on with Unger, he designed a syringe valve apparatus that is used in the transfusion from donor to patient by an unassisted physician became practical. So 1914, Albert Hustin introduced the use of sodium citrate as an anticoagulant solution for blood transfusion. However, the problem in this case is citrate toxicity because the overdue or the increased amount of the use of sodium citrate as an anticoagulant solution can create poison phenomena, which is your citrate toxicity, uh, which involves a different and complex cascade of cellular interactions, which is discussed in biochemistry, which we will not be diving into details. So 1915, Louis Lewison determined the minimum amount of citrate needed for anticoagulation and demonstrated its non-toxicity in very small controlled amounts. So moving on, 1916, Francis Python Roux and J.D. Turner introduced that glucose or have experimented on glucose, and glucose was tried as a preservative solution. They introduced the citrate dextrose solution for blood preservation. In 1941, Dr. Charles Drew, the director of the first American Red Cross blood bank at Presbyterian Hospital, the techniques he introduced the techniques in blood transfusion and blood preservation. So in 1943, John Luttig and Patrick Mollison introduced the formula for the preservative acid citrate dextrose or your ACD, which is up until now being utilized by most of the blood banks. So in 1957, Gibson introduced an improved preservation solution or a preservative solution called your citrate phosphate dextrose. Moving on, okay, Alfred von de Castello and Adrian Sturley, they discovered blood group AB, which is the rarest of the common ABO blood group systems. So Carl Senior and Alexander Weiner discovered the RH system. Yves Lapierre discovered the gel technology, which is up until now being utilized in most of the blood banks, actually all of the blood banks. Sosumoto Nagawa introduced antibody diversity. So moving on with the overview of basic genetics. So this time or for this particular slides, following slides, we will only be discussing not a discussion, but more on a presentation of your basic terminologies and processes. So, genetics is the study of inheritance or the transmission of characteristics or traits from parents to its progeny or offspring. So, recalling your basic cytogenetics or human biology. This is actually based on the biochemical structure of chromatin, which includes your nucleic acids and the proteins that constitute the genetic material, as well as the various enzymes that actually assist in the genetic processes, such as your replication. Father Gregor Mendel is the father of genetics, and he is very famous and known for his Mendelian's law of inheritance, which is actually comprised with three laws of inheritance. So, for the first law, is the law of segregation. It states that an organism or an individual contains two alleles and that these alleles segregate or undergo separation during meiosis, such that each resulting gametes will contain only one of the alleles from its parents. So it means that an individual actually contains two hereditary factors known as genes for any given trait. And when these gametes are formed, these two genes will actually behave like, let's just say, for example, particles and separate from one another, which means that the gametes that are formed will only have one of the pair of genes coming from its parents for particular trait. So moving on to your second Mendelian law, which is the law of independent assortment, which states that alleles for separate traits are passed independently from one another or of one another from parents to the progeny or offspring. So, lastly, for the third Mendelian law, which is your law of dominance, which is actually the most important of the three laws of Mendelian law of inheritance. So, it states that recessive alleles will always be masked by dominant alleles. Okay, taking for example, Gregor Mendel's experiment on pea plants. So, let's choose, for example, the height as the trait. Okay, whereas one trait is tall, given two capital letter T's, and the other trait short, which is regarded as two small t's. So Gregor Mendel actually experimented that when coupling or mating the pea plant with a tall trait with the short pea plant trait always produces an offspring of a tall trait. This is your first filial progeny. However, when he coupled two first filial progenies, he found that it resulted to a 75% of pea plants with tall traits and 25% pea plants with short traits, which accounts for a 3:1 ratio, right? So from this resulting experiment, he rationalized that when two different traits were coupled and one trait became evident on the progeny, doesn't literally mean that the trait unexpressed, which is the short trait, is gone permanently, but was only suppressed or inhibited by the expression um, by the more dominant trait, which is the tall trait. And from these concepts, we build the law of dominance. So moving on with several important terminologies in your basic genetics. When we say gene or genes, these are the fundamental units of heredity and the basic structural and functional units of genetics. When we talk about recessive traits, these are the traits unexpressed in a heterozygous condition, but expression will occur when it is inherited in a homozygous state. So, taking for example the one that we have previously mentioned in the law of dominance, for the short trait to be expressed, it needs to be inherited as two small t's because when paired with a dominant capital T, the expression of the short trait will be inhibited or suppressed by the more dominant capital T for the tall trait. So as opposed to your recessive trait, we have here your dominant traits, which are the traits that will be expressed even though inherited in a heterozygous condition, meaning to say inherited with or along with a small letter t. So when we say phenotype, these are observable properties of an organism. Genotype is the specific genetic constitution of an organism, and it is composed of the actual genes inherited from each parent. So when we say homozygous, we have two identical alleles for one or more genes. When we say heterozygous, we carry two different alleles for one or more genes. So moving on with the basic concepts of immunology and blood banking. So this is just a quick overview of your immune system. When we say immunology, so from the term itself, immunology, immune system, this is the study of the human immune system, primarily the response mechanism of the immune system or our body during an immunologic assault that is triggered by your pathogenic antigens that may present as polysaccharides, proteins, and so on. In relevance to the study, the field of medicine is actually in synergy in order to actually search, experiment, create, and manufacture different treatments for the disease caused by these particular antigens via studying our own immunologic response or how our body behaves with these particular antigens. While immunity, in its most basic definition, is the process by which a host organism protects itself from the attacks by an external agent or an internal agent or both agents. Specific immunity is a result of the exposure to a variety of agents capable of inducing an immune response. So we are talking about immunogens. For example, your vaccines and microorganisms that may colonize the body that that can cause disease states. So specific immunity may be cellular, which includes your T lymphocytes, B lymphocytes, and plasma cells, or humoral, which includes your antibodies and lymphokines. So specific immunity has three essential characteristics. We have the antigen, first, the antigenic recognition. So in blood bank and immunology perspective, this is the ability of the antibodies to distinguish or recognize self from foreignness or self from non-self or your immunogens or antigens. Specific reactivity is actually the ability of antibodies and as well as your lymphocytes to react with matching homologous immunogens encountered by the body. When we say, lastly, when we say immunologic memory, so it pertains to a person's antibodies, most particularly those that are sensitized with the antigenic material like your lymphocytes, and then to technically remember their homologous immunogen or their markers, and will show a strong reactivity against that particular antigen when it happens that a person have contracted the same antigen a long time after the first exposure. Next, a specific immunity may be acquired by natural or artificial processes, or specifically or particularly via naturally acquired and artificially acquired. So as for natural active, it includes the type of immunity that develops during convalescence, or in a more basic term, the type of immunity that we produce after the recovery from a disease state. So as for your natural passive, it includes the type of immunity that develops after the transplacental transfer of IgG molecules from mother to fetus during gestational period or pregnancy. Okay, for you, your artificially active includes the immunity after vaccinations, and artificially passive, the immunity produced via injection of gamma globulins for the induction of an immune state. Okay, this is a simplified tabular version of the differences between active and passive immunity. As to the sources, immunizing agents, a relative length of reactivity, effectiveness, and undesirable effects. So as for active immunity, the host is responsible for producing immune response. And as earlier mentioned, or from our previous slide, for natural active, it is induced after the recovery from a disease state, which will later on produce the immunity directed against that particular antigen that caused the actual disease. Artificial active is via vaccination, while for natural passive is your colostrum IgA as well as placental passage of IgG molecules from mother to fetus. So colostrum meaning it involves the mammary, which means to say that there is an IgA transfer during the breastfeeding stage in infants or newborns. So class, remember that your IgA is not only present on your serum, but also abundant in your body secretions like saliva, tears, semen, and so on. Colostrum as an example. So for the immunizing agent, or to induce an immune response on the active immunity is your antigen. While for passive, we have your antibody, which is derived from another source like the antibody transfer from mother to fetus. Active immunity provides a long life immunity, however, via a longer exposure to an antigen causing the particular disease or actual disease, while passive immunity provides the immediate protection. However, it will not last long as opposed to your active immunity. So take note that active immunity is more effective in adults while passive is effective in newborns. To conclude our table, we have the undesirable effects. So as for natural active, you need to experience the actual infection or the actual disease in order to sensitize your immune system to eventually produce an antibody specific for that particular antigen. So for passive immunity, there's what we call as the serum sickness, which will we will be broadened or which will be broadened or expounded into detail by your immunology subject. Specific immunity is mediated by two interrelated and independent mechanisms. So, humoral immunity, which actually involves your bursa or bone marrow derived B lymphocytes or your B cells, where your predominant antibodies are produced by your plasma cells. And this is your primary defense against bacterial infections. So take note, class, this is your primary defense against bacterial infections. Cell-mediated immunity primarily involves the thymus-derived lymphocytes or your T cells that can be initiated via direct cell-to-cell contact or via the soluble products secreted by cells, specifically your what we call as your cytokines. And this is your body's primary defense against viral and fungal infections, as well as or including tumor antigens or tumor cells, and graft rejection. However, specific immunity may not always be protective since it can also cause allergies like your famous hay fever or anaphylaxis. And the most clinically significant immune response is your autoimmune disease, where your immune cells targets or attacks your body's own self or your body's own cellular constituents due to the failure to recognize the these cellular constituents. For example, in conditions like thyroiditis, what else? Your systemic lupus erythematosus, and others to name a few. So for an additional information, we actually have two types of immunization. The active, which is actually intended as an immunoprophylaxis, meaning immunizing yourself with a response antigen to produce a response using homologous antigens, or in other terms, an immunity for future use. One best example is your COVID vaccination, polio vaccines, and so on. While the passive immunization is intended mainly or primarily for immunotherapies. So we have here is a summarized information about your cellular and humoral components of the immune system, starting with your first line of defense, which includes an intact healthy skin, mucous membranes, cilia, or particularly your ciliated epithelial cells that functions as. Okay, remember in your anatomy, it functions as brush borders and repels any foreign debris or molecules that may trigger a disease state. And cough reflex that can also be associated with your ciliated epithelial cells on your airways. So, okay, I will no longer dive into deeper or I will no longer dissect your complex anatomy since these are only overview information. Next, your biochemical factors, which includes your secretions and acidic pH of the vagina in normal physiology, which is actually enhanced by your normal microbiota Lactobacillus shirota strain, which is similar to your beverage Yakult. So moving on, also the acidity of the stomach caused by your parietal cells. And as I have mentioned a while ago, your secretions has abundance of secretory IgA molecules that is responsible for protecting your mucosal surfaces. Okay, next to your second line of defense, which includes your cellular and humoral. So we have here your phagocytic cells like your polymorphonuclear cells or collectively known as or your neutrophils, macrophages, and monocytes, including the very potent natural killer cells or NK cells. And take note, class, that in the case of monocyte macrophage, these are actually same cell types of phagocytic cells. When they are inside your blood vessels, they are called as monocytes. But if they migrate from blood vessels to tissues where they differentiate, they are called macrophages. So for example, your macrophage in your liver are called Kupffer cells. Macrophages in your placenta are termed as Hofbauer cells. And what else? Your macrophages that are present in your dendritic cells are called as your microglial cells, and so on. So let's leave the rest for your immunology class. So moving on with your humoral, which includes complement cascade, cytokines, and acute inflammatory reactions. So for the third line of defense, we have cellular lymphocytic origins like T helper cells, cytotoxic T cells, memory cells. For humoral, we have your antibodies, complement, and cytokines. Remember that one of the major functions of your immune system is the production of your soluble proteins called antibodies, which actually belongs to the class of proteins known as your globulins. Because, or why exactly? It is because of their globe-like protein molecular structure. So there was actually quite a story about this, its nomenclature. It was formerly known as gamma globulins due to its migratory properties in your gel electrophoresis. But in your modern immunology, or as of today, these are these are already collectively known as macromolecules or antibodies, or your most famously known as immunoglobulins. So these are complex protein molecules produced by plasma cells with specificity to an antigen that actually stimulated their production. So by physicochemical and immunochemical techniques, the or serologic techniques, the immunoglobulins consist of five different isotypes. We have your IgG, IgA, IgM, IgE, and IgD. IgG have gamma heavy chain, IgA have alpha heavy chain, IgM have mu heavy chain, IgE have epsilon heavy chain, and IgD have delta heavy chains, and they differ not only in these structures and their antigenic reactivity, or which means to say based on their serologic activity, and also with their carbohydrate content, but also in the size and their biological functions. IgG is the major immunoglobulin in normal human serum, which actually accounts for 75% of the total immunoglobulin present, whereas they are also the major antibody in a secondary immune response. Take note that IgG are the major antibody in a secondary immune response, meaning to say antibodies present during past infection. They are also the Ig class that can cross the placental barrier, which means to say that IgG is responsible for fetal protection and also, of course, your newborn protection during the first months of life. We actually have four subtypes of IgG molecules or antibodies. We have IgG1, IgG2, IgG3, and IgG4. Having IgG1 and IgG3, the best IgG subclasses in phagocytic or opsonization functions, and only IgG1, IgG2, and IgG3 can cross the placental barriers, so except for IgG2. Take note, class, that um, except for IgG3 that has a half-life of 7 days, all the IgG subclasses have a half-life of approximately 23 days, which is actually the longest of all Ig isotypes. And this is the precise reason why IgG is the best suitable isotype for passive immunization. Next is your IgA, which is actually present in the serum and various body fluids or secretions like the ones from the previous table, like your saliva, tears, colostrum, and so on. So take note that IgA can be oh, okay, it is a board exam pointer, class. IgA can be deactivated or inactivated by the IgA protease enzyme produced by your Neisseria gonorrhea. And since secretory IgA is the main responsible for the mucosal surface protection, deficiency to these isotypes will result to increased mucosal infections like atopy and certain several skin conditions. So moving on with your IgM, which is actually the most efficient immunoglobulin to fix the complement due to its pentameric structure, meaning it is the largest of the Ig subclasses or isotypes, which we will be broadened when we reach the ASG or anti-human globulin topic of in our blood banking lecture. So take note that IgM is the major antibody in a primary immune response. Additional board exam recalls class and pointer examples of IgM class antibodies includes your Wassermann antibodies in syphilis, heterophile antibodies in your EBV or Epstein-Barr virus, and lastly your rheumatoid factor. So in blood bank, um, we take note that IgG and IgM are the most clinically significant because of the thermal reactivity. Particularly your IgG thermal, it has a thermal reactivity at 37 degrees centigrade. That's the term warm antibody, and which is also capable of destroying transfused antigen positive RBCs, and your IgM isotypes that are reactive at 22 degrees centigrade or colder, thus the term cold antibody or cold isoagglutinin. IgE represents only 0.004% of total serum immunoglobulin and was actually originally called as reagent antibody. One of the unique characteristics of this isotype is being homocytotropic, which means that it has a very high affinity for the cells of the host or the person or an organism species that produce it, thus the term homo. Okay, most particularly for those tissues like your mast cells and basophils. And this is actually the main reason why IgE levels is elevated in the presence of allergic or anaphylactic reactions due to the nature of the mast cells and basophils that exhibits the ability to attack the skin cells, where the sensitization is localized or where the antigen is localized. So it is also the Ig subtype that increases along with eosinophils in the presence of helminthic infections. And finally, to conclude our Ig subtypes or isotypes is your IgD or delta immunoglobulin. It accounts for only less than 1% of the total immunoglobulin population, and due to its fragility, it can because it is very fragile in certain physiologic enzymes, it can be destroyed easily. So it is the least studied immunoglobulins, so its main function wasn't really definitive. So we have here your polyclonal and monoclonal antibodies, which are actually utilized in the form of commercially manufactured reagents used particularly in the detection of your non-agglutinating antibodies and for compatibility testing, which I will be discussing in a more complex and detailed discussion when we arrive at the anti-human globulin testing, or as we proceed with the succeeding week discussions. So, for the meantime, I'll be presenting you with the basic overview of both antibodies. So, for polyclonal antibody, these are actually antibodies produced in response to a single antigen with more than one epitope. And from the term itself, poly meaning it has different specificities and has a reactivity to multiple antigenic epitopes. And that's exactly why it is not specific. So as opposed to monoclonal, which is actually isolated from individual B cells from a polyclonal population and was actually propagated in a cell culture with hybridoma to produce a single antibody cell type from a single B cell type, which means that it is highly specific and well characterized and uniformly reactive. So this time class, we are narrowing down the overview concept discussion specific for your antibody types as a result to the exposure to your RBC surface antigens. So as for allo antibodies, they are produced after exposure to genetically different or non-self RBC antigens after blood transfusion, meaning to say the immunizing agent here is not present in the recipient. While autoantibodies are produced via sensitization to its own RBCs or antigenic determinants contained in its RBCs, which is present in conditions like your hemolytic anemias. And it actually has a wide thermal range like cold and warm reactivities. For example, uh, your cold paroxysmal hemoglobinuria and warm autoimmune hemolytic anemia respectively. So alloantibodies will result positive in the direct agglutination test, while autoantibodies can be removed via adsorption and elution procedures. Affinity, when we say affinity, it is the initial force of attraction that exists between a single Fab site on an antibody molecule and single epitope or determinant site on the corresponding antigen. And they are actually held via weak non-covalent bonds like your ionic bonds, hydrogen bonds. So what else? Your van der Waals forces and electrostatic bonds. While avidity is the sum of all the attractive forces between an antigen and an antibody. So meaning to say it is the measure of the overall stability of an antigen-antibody interaction or complex. And lastly, specificity pertains to antibody characteristic reactions like um, specific reaction, cross reaction, and no reaction, which in practical application, for example, during your blood typing, where the medical technologist is going to grade the strength of antigen antibody complex or reaction as to um, the specific reaction, cross reaction, or no reaction, which will be broadened when we proceed with your agglutination procedures in immunology and serology, and of course in your blood banking. Just like your cellular immunologic responses, your host factors are also identifiers of how well we or an individual can react to a certain pathogen or the degree of its ability to induce an immune reaction or response. Listed here on the left side of your screen is the complete list of factors that can affect an individual's immune system and immune response. We have your nutritional status, sorry, hormones, genetic inheritance, age, race, sex, physical activity level, environment, exposure, occurrence of disease or injury, which I will not be discussing into detail. And since these are quite objective and easy to apprehend, I will leave this as your reading assignment. And when we talk about tolerance, it means that when there is the presence of an active immunosuppressive response, it means, therefore, that there is a resistance that actually takes place that inhibits the initiation process for an immune response to take place. Thus, your definition, lack of immune response, which can either be caused by a naturally occurring condition or of any conditions like iatrogenic causes, like your a gamma globulinemia or hypogamma globulinemia, or experimentally induced for research and medical purposes. So, moving on with the RBC biology and preservation overview. So moving on, there are three areas or criterions for a normal erythrocyte survival and function. These are the following: your normal chemical composition and structure of the RBC membrane, hemoglobin structure and function, and RBC metabolism. And take note that the presence of defects in any or all of these areas will absolutely result in RBC surviving less than the normal 120 days lifespan in the circulation. An individual RBC has a diameter or span of 6 to 8 micrometers. It appears as salmon pink in color and has the characteristic biconcave disc morphology, also known as your discocyte morphology, which is also necessarily unique about an erythrocyte because it helps RBCs for a maximum surface contact of hemoglobin that greatly promotes your transport efficiency function of your RBCs for blood gas exchange functions. So let's take a further look at this following table. About 52% protein composition comprises your RBCs, which primarily are your integral proteins or transmembranous structures, and peripheral proteins that actually serves as your cytoskeleton for your individual RBCs. It mainly contains of your spectrin and actin structural components. And going back with your integral proteins, these structures actually serves as the transport and adhesion sites and signaling receptors that also functions to support the very important carbohydrate defined blood group antigens, or your immunodominant sugar molecules that characterizes your blood type. And one of the most significant constituent that is located on your integral proteins is your sialic acid or sialic acid, which is actually imparts a negative charge on your RBC surfaces or RBC membranes, which is responsible mainly for each individual RBCs to repel one another during circulation process. And the degree of the repulsion between individual erythrocytes is referred to as your zeta potential. So moving on, about 40% of your RBC membrane is composed of lipids, primarily constituting your outer membrane layers that includes your phosphatidylcholines, sphingomyelin, and glycolipids. And lastly, about 8% consists of carbohydrate structures. So this is an included nice illustration for anatomy visualization purposes, which shows your lipid bilayer structures, the cytoskeleton components, integral proteins, locations, and your peripheral proteins. You can find this on page 5 of the chapter 1 of your modern blood banking and transfusion practices 6th edition by Denise Harmening. You can also consult your Rodax for cross-reference. We have here two important characteristics of red blood cells that actually plays an important role, a very important role in their survival. So number one, deformability, which is promoted by the cytoskeleton structures or the triangular spectrin networks of an individual RBC. This actually promotes the flexibility and elasticity that basically gives the ability of the individual red blood cells to fold and become pliable when they move through very narrow blood capillaries or blood vessels during the circulation process, while in the presence of certain conditions that increases red cell ATP consumption, which of course declines its concentration in the red blood cells, and an increased membrane calcification due to calcium deposition are actually the manifestations of your spherocytosis, which has the hallmark of a decreased surface to volume ratio, and as well seen in elliptocytosis. And when we say calcification, it means that there is hardening that is taking place that will eventually cause your membrane rigidity and inefficiency of your individual RBCs. So when the spleen recognizes this phenomena or the changes that has occurred in your RBC membranes due to a certain condition, it will sequester that particular RBC, causing its destruction. So which will now affect its RBC, which will now affect your RBC survival rate. So number two is permeability. So your RBCs or red blood cells are permeable to water and anions, specifically your chloride and bicarbonate, while impermeable to cations like your sodium and potassium. However, in conditions that in the presence of conditions that increases permeability on ions, it will decrease or decreases your RBC survival. And how does it exactly happen? So I believe you are familiar with the term PISO, meaning potassium in, sodium out. So this pump mechanism is actually aided by your adenosine triphosphate or your ATP, and along with your sodium. So along with this pump, calcium ions are also pumped into the interior of your individual RBCs. And this energy-dependent calcium ATP's pump is actually modulated by calmodulin, which actually functions to prevent calcium buildup during the ionic pumps. However, if equilibrium is not maintained, that can be via certain iatrogenic causes or different several certain conditions, will result to a continuous ATP depletion, meaning to say more and more cellular energy is consumed in the process, resulting to a continuous decrease in ATP um, uh, decline, and a continuous deposition of calcium ions on the membranes of your individual RBCs, and more of the sodium being expelled out from the membranes. And we all know about the rule, where sodium goes, water goes. So meaning water is pumped out, so it will be pumped out along with the sodium ions, resulting now to a red cell dehydration, and will eventually result to membrane rigidity. And when the splenic cells again detects the changes in RBC membrane, these cells will sequester the particular individual RBC, resulting now to its decreased RBC survival. So that is what actually happens in your in conditions that results to membrane rigidity and cellular um, dehydration due to increased permeability to anionic compounds. So these are the four different metabolic pathways that an individual RBC utilizes in energy production. First is your Embden-Meyerhof pathway. This is the energy production via anaerobic glycolysis, which is actually the major pathway utilized by your red blood cells that accounts for 90% glycolysis in red cells. So next is your pentose phosphate pathway, or also known as your hexose monophosphate shunt. So in this pathway, reduced glutathione is made available via NADPH to prevent oxidative denaturation of hemoglobin. So what are the two hemoglobin prone sites of oxygen denaturation? We have number one, your iron in the heme ring, and number two, sulfhydryl group. Now, the oxidation of sulfhydryl group will actually cause your hemoglobin precipitation, which is also known as your Heinz body formation. So your G6PD class actually provides the only means of generating NADPH for glutathione reaction. So the absence of this, therefore, will result to RBCs to become more vulnerable to oxidative damage. So, and take note that G6PD deficiency is the most common human RBC enzyme deficiency worldwide. So moving on with your methemoglobin reductase pathway. This cascade of reactions maintains the iron present in the hemoglobin molecule in a functional reduced state, which is your ferrous ion, necessary, or which is actually important and necessary for oxygen transport. And lastly, we have your Leu-Behring-Rappaport pathway, which generates 2,3-DPG or 2,3-BPG that will enhance delivery of oxygen to your tissues or epithelial cells of your tissues. So how exactly? This is by decreasing your oxygen affinity or competing with oxygen for hemoglobin, which we will be explained in a more detailed manner on your hematology class. Okay, class. Since the main function of your RBCs is actually only to deliver oxygen to tissues and do not actually to consume it at all. So because we have learned from our biology class that RBCs are enucleated cells, and basically or literally does not contain mitochondrion to facilitate energy production. And this is the precise main reason why individual RBCs will undergo your anaerobic pathway, or your Embden-Meyerhof pathway, or non-oxidative pathway, or cascade of reactions to, or in order to produce energy via depletion of glucose molecules. So as you can see, these are the whole cascades of the four of your metabolic pathways. However, we will only be focusing on the major pathways, which is the Embden-Meyerhof pathway. So it is highlighted for a reason. So here is a nice illustration of my great, very old notes during my biochem teaching tutorial. Class, take note that in any form of medical sciences, this pathway is almost always present, and every inch of your body's cellular constituents or components undergoes in this process. So I will be dividing this collection of cascades into three main stages to make it more retention specific and based from the goal of each stages or the products that are formed by each stage. So as for stage one, we basically prepare the glucose for breakdown, meaning we trap these molecules inside the cytoplasm of the RBCs and transforms this low energy glucose into unstable high energy glucose molecules. So in stage 2, we actually use these unstable high energy glucose molecules that were formed during your stage 1 and basically breaks them down into two carbon molecules needed for your stage 3 to take place. And eventually, in stage 3, we harvest the energy within these molecules and we produce now your energy, which is your adenosine triphosphate or your ATP molecules. Okay, beginning with your stage one. In here, we actually transport the glucose molecule into the cytoplasm, or uh, we take the glucose molecule from the extracellular environment across the plasma membrane into our cytoplasm and transforms this glucose molecule into glucose fixed phosphate or G6PD via the enzyme hexokinase, or since it is inside already inside the RBCs, we call this, or we can call this, hemokinase. So we basically extract one negatively charged phosphate molecule from the ATP and transfers it to or attach it to our glucose molecule, forming your G6PD. And this transfer of phosphate group is what we call as, okay, a recall, a phosphorylation. So imparting our glucose with a negative charge, which is a negative phosphate group. And this is actually the mechanism that traps the glucose molecule inside the cell and prevents it from exiting the cytoplasm. And it will be further destabilized via the action of the enzyme glucose phosphate isomerase, transforming it into fructose 6 phosphate, which does not require energy for the cascade to take place. And with the use of phosphofructokinase enzyme, we require an ATP molecule to basically initiate the conversion of fructose 6 phosphate into fructose 1,6 biphosphate, which is now your unstable high energy molecule. Okay, take note, class, however, that when our body contains an ample amount of energy, the conversion of your fructose 6 phosphate into fructose 1,6 phosphate will not take place. And the fructose 6 phosphate will be stored as your glycogen that will be used as an energy stored molecule for future energy use. But if the body is in need of energy, the cascade will take place and continues in your stage 2. So in stage 2, class, we take the unstable high energy molecule from stage 1, which is your fructose 1,6 biphosphate, and transforms it into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, or your PGAL, via the action of your aldolase enzyme. However, what is actually needed to proceed with the stage 3 of your anaerobic glycolysis for energy production is two carbon molecules. So that it means to say that we actually need two PGAL molecules to undergo or to continue the cascade to our stage 3. So that's exactly why with the use of the triphosphate isomerase, we basically transform the dihydroxyacetone phosphate into one more molecule of glyceraldehyde 3-phosphate, which now forms our two carbon molecules, which is your two PGAL molecules or two glyceraldehyde 3-phosphate molecules. So now, your PGAL molecules are reacted via glyceraldehyde 3-phosphate dehydrogenase that basically uses your NAD as a cofactor. So in this case, we basically or actually extract a hydrogen molecule from the PGAL and attaching it to your NAD forming NADPH, and the removal of the hydrogen molecule from the PGAL will result to a phosphate group to be attached with your resulting molecule, which is 1,3-diphosphoglycerate or 1,3-DPG or 1,3-D 1,3-BPG. And now, with the use of your phosphoglycerate kinase, will remove the attached phosphate group or molecule from the earlier cascade and returning it back to your ADP to form your first ATP molecule, which now forms your 3-phosphoglycerate. So in this following cascade, the enzyme phosphoglyceromutase will transform three um, will transform your 3PG into 2PG, and this process will actually initiate the addition of a hydrogen molecule to the terminal oxygen of your 2PG, and this process will actually produce now a H2O or a water molecule and phosphoenol pyruvate. And continuing with the cascade class, pyruvate kinase will initiate the transfer of phosphate from phosphoenol pyruvate to your ADP, forming now your second ATP molecule and forming your final product of the Embden-Meyerhof pathway or an anaerobic glycolysis pathway, which is your pyruvate. So, okay, class, remember that on your second stage or stage two, we actually produce two molecules of PGAL, which exactly means that your stage three actually produced two NADH, two ATP, two ATPs, and two hydrogen water molecules. So, this is the process or processes as to how your RBCs produces energy for the body, and the basic knowledge and mastery of this cascades will actually aid you to understanding the succeeding concepts that all succeeding concepts that will involve uh, about your RBCs or red blood cells. Okay, class, in this particular case, in this particular slide, I will only be discussing a rough discussion about your oxyhemoglobin dissociation curve, since it will be discussed into a detailed manner or into a detailed discussion by your hematology class instructors or professors. So shift to the left curve of your oxyhemoglobin dissociation curve is when when your hemoglobin molecule has an increased affinity for oxygen and does not readily release it to your epithelial cells of your corresponding tissues. So actually, certain environmental conditions can actually cause this left shift effect on your oxyhemoglobin dissociation curve. For example, it actually includes your decrease in body temperature, a decrease in 2,3-DPG or 2,3-BPG, or decrease on partial carbon dioxide concentration, and an increase in your blood pH. Okay, all of this mentioned actually increase the affinity of hemoglobin for oxygen. Thus, or it means to say that there is a potential decreased delivery of oxygen molecules to your epithelial cells of your corresponding tissues. One example of such is in the condition during hyperventilation. So in cases of hyperventilation, in which larger than normal amounts of carbon dioxide are lost as the individual breathes excessively, often because of severe anxiety and other causes, for example, nervousness, hypertension, and so on. This carbon dioxide loss causes an increase in pH, of course, okay. Um, The hemoglobin binding of oxygen causes it to release free hydrogen ions, which eventually reduce the pH to normal and shift the curve back to its normal position. So the oxygenation of your hemoglobin also causes it to release 2,3-DPG, resulting now to a decreased level of erythrocyte 2,3-DPG and allowing for further oxygen binding. So going back, shift to the left is when there is a shift to the left on the oxyhemoglobin dissociation curve, we are actually in the relaxed form of your hemoglobin structure. So moving further with your shift to the right curve on your oxyhemoglobin dissociation curve. So a shift to the right actually represents a decreased affinity of hemoglobin for oxygen, and this may actually result from an increased body temperature, increased concentrations of your 2 to 3-DPG or partial carbon dioxide, or decreased blood pH. All of this mentioned or actually increases the delivery of oxygen by your hemoglobin to your tissues of your or epithelial cells to your corresponding tissues. Example condition in physiologic state during exercise in which the muscle tissue temperature rises, or in cases of abnormal buildup of hydrogen ions in your present in your renal failure, particularly in severe cases of diabetes and so on. So the acidic state actually results to excess hydrogen ions that are released into the blood. These ions can be bound by non-oxygenated hemoglobins. So as prescribed by your Bohr's effect on your pH, or just a recall, also a recall from your physics, the Bohr's effect, which actually regulates the relation between pH and hemoglobin oxygenation, where hemoglobin must release its oxygen to the tissues before it may bind with the hydrogen ions. So by binding with these ions, the hemoglobin actually plays a role as a buffer, and eventually the pH is brought back to your normal physiology. It means therefore that eventually it will cause the shifting the curve into the direction of normal oxyhemoglobin dissociation curve. So, okay, class, for the rest of the succeeding slides or presentations, I will only be introducing you to the overview concepts of each of the following topics. So, since we will be giving a more complex and detailed manner of discussion as we move through the week meetings and week discussions. So, RBC preservation to provide a viable and functional blood components for patients requiring blood transfusion, we actually perform or utilize RBC preservation. So, the goal of this is to provide a viable and functional blood components before or prior to blood transfusion. We have two criterions in evaluation of new preservation solutions and storage containers. We have RBC survival greater than 75% over 24-hour post-transfusion. Free hemoglobin must be less than 1% of the total hemoglobin. RBC viability is a measure of your in vivo RBC survival following your transfusion practice or procedure. It determines or measures the post-transfusion RBC survival in vivo. So FDA approved, it should be greater than 75% RBC survival, and critical ill is less than 75% RBC survival. So how is it done? RBCs are actually taken from healthy subjects or um, subjects with or person individuals with no hemolytic anemias or other blood diseases. They are stored and then labeled with radioisotopes, reinfused with the original donor, and measured 24 hours after transfusion. So, despite the FDA requirements, the 24-hour post-transfusion RBC survival is actually at outdate can be less than 75%. So, take note of that. So, take note also that 1 to 6 degrees centigrade is the optimum temperature for RBC viability. So, the loss of RBC viability has been correlated with the storage lesion of the storage or the adverse effects associated with prolonged storage of blood. Okay, listed here in this particular slide are the biochemical changes associated with RBC storage lesion. So, okay, class, when you say our storage lesion, we are referring to the adverse effects associated with prolonged RBC storage. So, the viable cells, glucose, the adenosine triphosphate, lactic acid, pH, and 2,3-DPG are the parameters, also the oxygen dissociation curve, the plasma potassium, and plasma hemoglobin. So, viable cells, glucose, ATP, your pH, and 2,3-DPG um, are decreased. These are the biochemical reactions. They are decreased. While for your lactic acids, your plasma potassium content and plasma hemoglobin are increased on prolonged RBC storage. So your oxygen dissociation curve, it will shift to the left since the increase in hemoglobin and oxygen affinity is prominent or evident and is observed and is initiated by the prolonged RBC storage, and also less oxygen delivered to the tissues. So here are the lists of approved anticoagulants preservative solutions for whole blood and RBC storage at 1 degrees centigrade to 6 degrees centigrade. So as you can see, adenine increases ADP levels, thereby driving glycolysis towards the synthesis of ATP. So it means to say that in CPDA1, with the presence of adenine, your the action of your adenine actually initiates or your forces your glycolysis to take place, creating more and more ATP. So and that's exactly why above all of the among the four of the approved anticoagulant preservative solutions, CPDA1 has the longest storage time or days, which is approximately at most 35 days of storage. So for your acid citrate dextrose formula A, your CPD and CP2D only have a storage days or duration of 21 days. So take note, class, review. Remember that we have here four approved anticoagulant preservative solutions. You have ACD, A, CPD, CP2D, and CPDA1. So the first three have a storage span of only 21 days, and CPDA1 has 35 days because of the presence of adenine that initiates the glycolysis process. So that's why it will provide more ATP molecules for your red blood cells for them to stay viable for at most 35 days. Okay, class, here is a complete list of the various chemicals used in anticoagulant solutions and their functions during the storage of red blood cells. So, as previously mentioned, that your CPDA1 contains adenine, which actually makes it or facilitates the longer duration of a storage time from 21 to 35 days of or the shelf life of your RBC viability. So we have here the chemical components, the citrate or the sodium citrate or citric acid, the monobasic sodium phosphate, and the dextrose, including your adenine. So as previously mentioned, CPDA1 contains adenine, and your ACDA, CPD, CP2D contains, as well as your CPDA1 contains all of these mentioned chemicals and an addition of adenine. So the function of your citrate is to chelate the calcium that further prevents, or it means that it prevents the clotting of your RBCs while on storage. So monobasic sodium phosphate maintains pH during storage, and it is actually necessary to maintain adequate levels of 2,3-DPG for your red blood cells while undergoing the storage span of over 21 days. So dextrose is a substrate for ATP production, which is required for your RBC's cellular energy. Okay, moving on with your additive solutions. So these are the solutions added to your RBCs after the removal of plasma with or without platelets. And due to the removal of plasma components, much of the nutrients needed to maintain RBCs during storage are also removed. So as for the benefits of RBC additive solutions, number one, extends the shelf life of red blood cells up to 42 days by adding nutrients that are actually necessary to increase the viability of your RBCs during the storage. It also allows the harvesting of more plasma and platelets from the unit. And lastly, it produces an RBC concentrate of lower viscosity that is easier to infuse or transfuse for in blood transfusion. So we remember that the characteristic of our red blood cells is viscous. So the action of this additive solution is to lower its viscosity and makes it easier, or it creates it, or it transforms it into a lower viscous manner or state for it to be transfused easily during blood transfusion. So currently, three additive solutions are licensed in the United States. We have your Adsol, manufactured by Phenol Incorporation, Nutricel by Haemonetics Corporation, Optisol by Terumo Corporation, and Solex. So these are the abbreviation: AS1 for Adsol, AS3 for Nutricel, Optisol for AS5, and Solex for AS7. So they have the same storage durations, which is 42 days. We will not be diving into specifics or magnifying or dissecting every detail of these additive solutions, since there is a separate week discussion for their proper or detailed explanation and discussion. Okay, moving on with RBC freezing. This is primarily used for autologous units and the storage of rare blood types, for example, A negative, O negative, AB negative, and so on. Glycerol is the most commonly used cryoprotective agent. Negative 65 degrees centigrade is the usual storage temperature when performing the RBC freezing procedure. We have two different glycerol concentrations. We have the 40% weight over volume, which is a high concentration glycerol, and 20% weight by volume low concentration glycerol. So most blood banks uses this high concentration glycerol. And okay, and just for an additional information, there has been an experiment or a blood bank experience that has shown that a 10-year-old or 10-year storage periods do not actually give adverse effects to your RBC viability and function. So that is how efficient your RBC freezing is. So, okay, class, we are still on RBC freezing. So, defining deglycerolization, it is the removal of glycerol from thawed and previously frozen RBCs before or prior blood transfusion. So, glycerol, we have mentioned a while ago that glycerol actually acts to protect RBCs during freezing and thawing, but it can also cause hemolysis if not removed or washed out before or prior to blood transfusion. So the removal of glycerol is actually achieved by systematically replacing the cryoprotectant with decreasing concentrations of saline. So the usual protocol actually involves washing with salines of three different concentrations. We have first washing with 12% saline, followed by 1.6% saline, with a final wash of 0.2% dextrose in normal saline. So take note, class, that after the glycerolization, the blood unit must be transfused within 24 hours after thawing, because the blood unit has been processed in an open system, meaning it has already been opened, and the glycerolization or the glycerol has been removed from the blood cells, meaning to say it will be prone to contaminants, deterioration, and so on. So it must be transfused within 24 hours after thawing. So we can put this thawed or the glycerolized blood unit in a 1 to 6 degrees centigrade temperature while waiting for the blood transfusion, and take but let us remember that it should not stand inside the um, freezer for more than 24 hours, since it will be deteriorated and will not be viable for blood transfusion. So next we have the advantages of high concentration uh, glycerol technique over low concentration glycerol technique. So the initial advantage, we have the initial freezing temperature of negative 80 degrees centigrade for high glycerol, and 100 negative 196 degrees centigrade for low glycerol. So in um, high glycerol, there is no need for control freezing rate, as opposed to your low glycerol that requires the need to control of the freezing rate. So the type of freezer required for high glycerol is a mechanical type, while low glycerol is liquid nitrogen. The maximum storage temperature for high glycerol is negative 65 degrees centigrade, as opposed to your 120 negative 120 degrees centigrade for low glycerol. The shipping requirements for high glycerol only requires dry ice, compared to your low glycerol, which requires liquid nitrogen, which is more expensive than your dry ice. So the effect of changes in the storage temperature. So under high glycerol, it can be thawed and refrozen, while in low glycerol, it is critical. It means to say that this procedure can easily deteriorate the blood unit. Next is RBC rejuvenation. This is the process in which your adenosine triphosphate and 2,3-DPG levels are restored or enhanced by metabolic alterations by or via addition of your Rejuvesol, PIPA, and others, to name a few. So your Rejuvesol is manufactured by Ensight Systems. This is the only FDA approved rejuvenation solution in the United States. Your PIPA, meaning phosphate, inosine, pyruvate, and adenine, which are necessary um, factors or solutions or constituents that are highly required for RBC rejuvenation. So RBCs are stored in the liquid state and can be rejuvenated at outdate or up to 3 days after the outdate. So these concepts will be broadened during your component preparation uh, on the allotted week discussion. And finally, your platelet preservation, which I will not be, of course, just like the other topics, will not be diving into details. So here are the quick overview and the general concepts only. So the normal platelet count is 150,000 to 450,000 per microliters. It is released and circulate approximately 9 to 12 days as small disc shape with average diameter of 2 to 4 micrometers. So they are stored or can be stored at 20 degrees centigrade to 24 degrees centigrade or room temperature for 5 days with continuous agitation. So the major concern associated with the storage of platelet is the potential for bacterial growth. If prepared platelets contain bacteria because of the contamination during phlebotomy, or if the donor has unrecognized bacterial infection. So that is the main risk, or the major concern that is associated often or frequently associated in blood banks with pertains to platelet preservation. So here, to end up our class discussion, I will be leaving you with multiple several trivias. A person has 5 to 6 liters of blood in their body, and for 150 or 500 ml is the amount of blood a normal person can donate. About 63 ml or 70 ml is the amount of anticoagulant used during blood collection. In Of course, guys, we are pertaining to the blood bank or immunohematology. So a person can donate blood every 8 weeks, and the body recovers the blood very quickly, as fast as 24 to 48 hours to in order to recover blood plasma, and about 3 weeks for red blood cells, depending on the um, state of your bone marrows and precursor cells. And for platelets and white blood cells, they are recovered within minutes only. So that is the end of our discussion. Thank you for your attentiveness, time, and diligence. So for our most of the topics that were not discussed in a detailed manner, okay, we will be having separate lectures for each of these topics. However, since this is only on week one, um, it is okay for to have this kind of light discussions. Our discussions proper will begin next week on your ABO blood group system. Once again, thank you and have a nice day.